All right, I think we'll get started. Welcome everyone to our ACTISting webinar, and delighted to have our presenter today, Ian Master from the NHMRC Clinical Trials Centre at University of Sydney. I'd like to begin by acknowledging the traditional custodians of the lands on which we are all gathered today. For me, it's the Bunurong people of the Kulin Nations, and pay respects to their elders, past and present, and also those of all the lands on which you're all meeting. So I'm delighted to welcome Ian Marshner for giving our talk today, and he's going to talk about historical and non-concurrent controls in clinical trials. So Ian's a Professor of Biostatistics at the University of Sydney and Director of Biostatistics at the NHMRC Clinical Trials Centre. He has many years of experience, over 30 years of experience, in fact, working on clinical trials and methodological research. He is a chief investigator of AUSTRIM, Australian Trials Methodology Research Network, and previously he was head of statistics at Macquarie University. Before that, he was director of biometrics at Pfizer Pharma, and before that, even associate professor at Harvard. So a very wonderful pedigree there. So very much looking forward to hearing about non-concurrent controls and new ways of looking at them from Ian. So I'll hand over to you, Ian, to share your slides.
Thanks, Andrew. That should all be sharing now.
Yeah, that looks good. I should mention, we'll leave questions for the end, but please feel free to ask any questions using the Q&A button, and it should be in the bottom right-hand corner of your screen.
Okay, thanks very much for that introduction, Andrew. And I would like to point out I've got the same number of years of experience as Andrew does, as he pointed out earlier today. So welcome everyone to this seminar on historical and non-concurrent controls in clinical trials. As Andrew alluded to, I'm a biostatistician. This talk won't be very statistical. It's more, I guess, about some of the concepts associated with historical and non-concurrent controls, and particularly around some of the issues with interpreting randomised and non-randomised comparisons in that context. And that's really one of the reasons why historical and particularly non-concurrent controls is an important methodological area, because it does get us into the realm of non-randomised comparisons. And as we all know, the gold standard for comparative trials is a randomised comparison of our experimental intervention with a standard of care, which is the control group, whereas historical and non-concurrent controls take us into the realm of non-randomised comparisons. And so we'll delve down into what we mean by that, and particularly try to explain and define some of the concepts that underlie the use of these particular types of controls. Before we get into some of the definitions and discussions, just to give us a concrete introduction to what we're talking about here before we delve more deeply into it. When we talk about historical controls, we're thinking of participants in a study who we have taken from external to the current trial and hoping to use as a control group for our current trial participants. And as well as being external to the current trial, they would typically be from an earlier point in time, so some sort of earlier study or other type of data set. Whereas non-concurrent controls come from within our current trial, but are not concurrently randomised within that trial. And I've used that term concurrently randomised. That opens up a whole sort of treasure chest of different ideas and concepts that I'm currently just glossing over, but part of what we'll be doing today is to delve into that in more detail. Why are we interested in these sorts of control groups when we know that randomised control groups are the gold standard? Well, it's really a bias information trade-off. So from the plus side, historical and non-concurrent controls can increase the amount of information that we've got. So they can allow us to have more power, shorter confidence intervals, these sorts of things. And this may be quite advantageous, particularly in situations where it might be difficult to recruit participants to the study. So in rare disease contexts, for example, or even in earlier phase trials. So that's the reason why people might be interested in using historical and non-concurrent controls. But the flip side is the potential for bias, and in particular, the potential for there to be systematic differences between the control and intervention groups for various reasons, but one particularly common reason is because of trends over time in the background risk. So, for example, the risk of patients having an event may be changing over time, and that can mean that there are systematic differences between our intervention-treated patients and those in the historic and non-concurrent control group. Quick example of that, this was an example that was published a couple of years ago. This is a hypothetical example, but just in relation to the use of non-concurrent controls, in this case from platform trials, and we'll talk a little bit about the different sorts of trials that yield these types of controls, but this narrative was sort of crafted in the context of platform trials. And the point that's being illustrated here is that if we have a trend in background risk, and in this case, we're talking about mortality from COVID-19 in hospital, and in that context, it's reasonable to postulate that treatment might be getting better over time. We might be protecting patients against mortality to a greater extent over time. So if we look at mortality in two different time epochs, in this case, April and May, where the mortality is reducing over time, a group of controls recruited in this period will have a lower mortality than a group recruited in this period. And collectively, they will have a mortality that's somewhere in the middle, an average of those two. If we now conduct a study of a new intervention and only do that in May, for example, then what looks like a nice treatment difference of 5.9% versus 9.3% is actually due to a spurious treatment effect that is caused by the non-concurrent controls from this April period having a higher risk. And so that's an example of how non-concurrent controls could introduce bias due to systematic differences, in this case, in background risk. There are many other potential reasons why those systematic differences might exist as well. All right, so what are some of the typical studies that yield these types of controls? We just alluded to platform trials. For non-concurrent controls, the typical types of studies that yield such control groups are these, particularly these innovative new designs, so platform trials, adaptive trials, multi-arm, multi-stage trials. These are all designs in which, in some way, the treatments may be changing over time, or the way in which participants are allocated to treatment may be changing over time. And that's the source of the non-concurrent control groups. In these sorts of studies, typically, we would have both concurrent and non-concurrent controls in the same study, although for the purpose of comparing between experimental groups, we may even have fully non-concurrent comparisons there. And the example that I've given here is the STAMPEDE study, a well-known study in metastatic prostate cancer. It uses what's called a multi-arm, multi-stage design. And the non-concurrent controls in this case arise from the fact that treatments are being added throughout the study and also dropped throughout the study. So the treatments are not being recruited over the same period of time. And so the treatment A at the top there is the control group. If you look at, for example, a treatment such as treatment G, which happens to be abiraterone added to the standard of care there, that's recruited over a much shorter period of time. So all of those controls in group A that were recruited outside that period of time would be what we call non-concurrent controls. And we'd be faced with the question of whether we should use them or not in the assessment of that treatment G, or whether we should just restrict ourselves to the randomised concurrent control groups. From the point of view of historic control studies, a couple of different ways in which we might bring in historical controls, which, as I said, tend to be external to the current study that we're conducting. One approach is that we have a single arm study that we are conducting, let's say an early phase oncology trial, where this is quite common. And we augment that single arm study with a control group from a previous clinical trial, or possibly even an observational study from a similar population. So that would be a common historical control situation. Another situation is where we do have a randomised controlled trial with a randomised control group, but that control group might be too small for adequate power. And so what we might consider doing is augmenting that control group from a previous trial or observational study, again, in a similar population. Certainly of those two, type 2 is the preferable one, because what we have there, even though we have an inadequately controlled study, we do have fundamentally a randomised reference group that we can use, and that's a very powerful feature with which to assess the appropriateness of bringing in historical control. So of those two sorts of historical control studies, type 2 is definitely better. Now, before we start getting into the sort of why and how of these historical and non-concurrent controls, I'd like to just spend a few slides delving into what we mean by randomised and non-randomised comparisons, because it's so fundamental to interpreting the validity of using historical and non-concurrent controls. Some of these slides may seem a little basic to start with, but I guess what I've found is that once we delve into these non-randomised comparisons, there is quite a bit of nuance around these seemingly familiar concepts that is not present so much in the sort of traditional randomised control trial context. So I'm going to go through, I guess, to build up the narrative through some very familiar concepts. Obviously, we're all familiar with randomised controlled trials. And typically when we conduct a clinical trial, that's the design that we're using, the experimental design that we call a randomised controlled trial, which has a number of familiar features to it. It's conducted in a specific population defined by eligibility criteria. It has a control group, which is what we're discussing in depth today. Obviously has an intervention group or even groups that we're interested in assessing. It has allocation to these treatment groups using randomisation, ideally with blinding. Today we're not talking too much about blinding, but that's an equally important aspect to the randomised control design. And then, of course, prospective follow-up and comparison between the groups. And so what I want to do is just delve in a little bit more to what we mean by the control group and the allocation of groups using randomisation, which are fundamental to interpreting non-concurrent controls. So the control group, of course, is a group of participants who did not receive the treatment or intervention that we are studying. And the purpose is for the control group to provide a benchmark for assessing that intervention. So typically a standard of care, which could even mean no intervention. And the key thing is that the control group needs to be representative of what would have happened to the intervention group if they had not received the intervention. And so in that way, it has to be chosen in a way that it's representative of the same population as the intervention group. And this is where the complexity comes in with historical and non-concurrent controls, because there's a potential for that representativeness to break down. And the reason for that is, of course, randomization. So randomization is what provides us with that representativeness and validity for the comparison between the intervention and the control group. Of course, and I guess over the next two slides, I'm going to take what we probably all assume is a fairly simple concept and just show that when we have non-concurrent controls and historical controls, it becomes a bit more nuanced. Randomization, of course, is allocation to the treatment group at random using some sort of chance mechanism. Its purpose is to produce an unbiased, or what we might think of as fair, comparison. Its key positive motivation is that it allows estimation of the causal treatment effect. So in other words, it allows an assessment of the causal effect of the treatment in protecting whatever it is that we're trying to protect in the participants. And equally importantly, it provides a basis for statistical inference because it allows us to understand the sampling variability and the statistical properties of our measures of the treatment effect. As well as that, on average, it ensures that the two groups are identical, with the one exception of the intended intervention. So that's all something that we probably assume we kind of understand and know. But it's when we come to a randomized comparison and defining what we mean by a randomized comparison that we start to run into some complexity when we have non-concurrent controls in our study. So a comparison, so given that we have just defined what we mean by randomization, a randomized comparison therefore, is a comparison of the intervention group to the control group where the treatment has been allocated using randomization. And so that's where we can get our assessment of the causal treatment effects. The complexity, though, arises in actually defining what we mean by a randomized comparison. And in particular, allocation of the intervention using a chance mechanism is not a sufficient condition for defining what a randomized comparison is. Just because the treatments have been allocated at random doesn't mean that we have a randomized comparison. And in particular, we have this concept of what we might think is somewhat of an oxymoron, a non-randomized random allocation. We can have allocation to the treatments at random, but the associated comparison that is yielded from that random allocation is a non-randomized comparison. And it's important when we come to interpret information from non-concurrent controls that we understand that the difference between a non-randomized and a randomized allocation and comparison. And we're going to spend a bit of time defining what we mean by a non-randomized comparison and therefore effectively a randomized comparison. But just to give a top-line version of where this oxymoron, if you like, is coming from, for a comparison of a control group and a treatment group to really be considered a randomized comparison, it's not sufficient for there to have been a chance mechanism. We additionally have to have all participants being assigned to the control or treatment using the same chance mechanism. And in a traditional clinical trial, this distinction doesn't typically arise because there's only one chance mechanism arising. But once we have some of these more elaborate designs, we can have multiple chance mechanisms being used in the same study. And those multiple chance mechanisms lead to the occurrence of non-concurrent controls. So let's discuss some examples and nuances of this non-randomized random allocation, which is really something, I guess, that applies more so to non-concurrent controls than historical controls. For historical controls, it's kind of easy to see that we have a non-randomised comparison there because there's no chance mechanism involved in the historical controls. But for non-concurrent controls, the situation is somewhat more subtle because, as I said, we can often have multiple chance mechanisms operating, and those chance mechanisms in their entirety do not necessarily provide a randomised comparison. One of the interesting things about that I've found sort of working over the last couple of years in the area of non-concurrent controls is that it's a concept that's commonly discussed, particularly in relation to some of these newer, more novel designs, the platform designs, MEMS designs, adaptive designs, this sort of thing. But it's a concept that hasn't really been unambiguously defined. So there is, I guess, an assumption that underlies the terminology non-concurrent controls. And that assumption typically is that we're talking about controls randomized at a different time. And in fact, what I'll try to present to you is the viewpoint that that's not sufficient to capture the full range of possible situations in which we have non-concurrent allocation methods and hence non-concurrent controls. And that really what we need is a definition that's based on randomization method rather than time. And that's partly what I'm going to talk to you about as well. So some examples. Well, they're the sorts of examples, I guess, that I gave earlier around the occurrence of non-concurrent controls. Adaptive studies, particularly where we have adaptive randomization, provide examples of non-randomized random allocation in the sense that with adaptive randomization, we have parallel groups of patients potentially. We may not even have the treatments changing over the time, but what is changing is the chance mechanism that we're using. For example, we might have the randomization probabilities changing over time. And so patients being randomized under these adaptive randomization schemes do not all have the same chance mechanism applying to them, and therefore that leads to non-concurrent controls. With platform trials, the adding and the dropping of treatments, as I said earlier, leads to non-concurrent controls because some patients will be randomized under a 2-treatment design, other patients under a 3-treatment design, etc. And some of the flexibilities of these novel designs lead to non-concurrent controls as well. So eligibility differences. Maybe not all participants are eligible for all treatments. So some patients may be randomized to a different collection of treatments than other patients. Site differences. Maybe not all sites offer all treatments. And there's a fascinating new design, actually, which we could have a whole talk about, which is the practical design, where patients may not even consent to all treatments. So patients may be in a position to consent to some but not all of the treatments, and therefore they're randomized under different chance mechanisms according to what treatments they consent to. Just to give some concrete examples from real trials, firstly, I'll give the example of the ASCOT trial, which is the Australian COVID trial run out of the Doherty Institute. And it was an adaptive platform trial that studied a range of treatments, but the domain that I'll talk about here is the use of anticoagulation therapies for non-critical hospitalised COVID-19. And in this particular design, what we had was 4 treatments. So the control treatment is low-dose anticoagulation, low-dose heparin. And then we have intermediate dose and therapeutic dose plus the low dose combined with aspirin. So 4 treatments altogether with the control being the low dose. And in this particular design, there were various randomization schemes operating due to such factors as not all patients being eligible for aspirin. So therefore, those that were eligible for aspirin initially were randomized between 3 treatments, whereas those who were not eligible for aspirin were randomised between 2 treatments. And what that means is that the middle purple group here, which is a control group of patients, is not a group of concurrent controls for this pink group. Even though they were randomised within the same time frame, this is a non-concurrent control group because they're not randomised under the same chance mechanism. And likewise for the therapeutic dose heparin, this group of control patients here is not a concurrent control group for this therapeutic dose group of patients. The concurrent controls come only from this group here. And so this is an example of one of these platform trial designs yielding concurrent and non-concurrent controls within the same design. Another example here, this is from the REMAP-CAP trial that many people will know, which is also COVID-19, but for critical COVID-19. This was this is a schematic that relates to the simvastatin domain of REMAP-CAP. Now, in this particular domain of REMAP-CAP, we have parallel randomization to just 2 treatments, either statin, simvastatin in the red, or no statin, in fact, open label in the green. So we have a parallel group design, but nonetheless, we do have non-concurrent controls because over time, the chance mechanism is changing. So the randomization to the 2 groups changed from 50-50 to 90-10, and then back to 75-25 over time. So the entire collection of green controls is not a concurrent control group for the entire collection of red experimentally treated patients. In fact, the concurrency only comes from within what I've called the stages here, where the chance mechanism remained the same. And so this is, again, an example in which we can have random allocation to the groups, but not yielding a randomized comparison in its entirety. We only have randomized comparisons within the stages where the chance mechanism remained the same. And we'll come back to how we therefore unravel and interpret that non-concurrency when we come to the analysis. The effects of this non-randomized allocation can be seen usually in the baseline characteristics. So this is the baseline characteristics table for the full cohort of the Ascot study. And we see a number of sort of really quite substantial imbalances that's caused by the non-concurrent controls within the overall groups. Vaccination status differed from 15% to 54% over the 4 treatments. The countries obviously differed, and age from 48 to 58, that sort of thing. So that's the I guess the practical consequence of these non-concurrent controls or non-randomized allocation, and identifies the need to sort of unravel from that information the randomized information. I'm going to come back to this table and show you how we can do that unraveling in a minute as well. All right, so I'm gradually moving towards now what I might call a rigorous definition of non-concurrent controls, which will help us to understand how we should deal with them. Before I get to that, I want to introduce one important key concept that's required in that definition and in interpreting non-concurrent controls, and that's the concept of a concurrently randomized cohort. And I guess I've given you a little introduction to that in terms of for the Ascot and the REMAP-CAP study, identifying those groups that were concurrently randomized, and that's going to be key to this definition of what I call a concurrently randomized cohort. So the definition of a concurrently randomized cohort is a subgroup of participants who all had the same treatments available to them, and they were all allocated to one of those treatments using the same randomised allocation method. So the same randomisation probabilities, stratification factors, etc. So within a given study, within a traditional fixed design study, we only have one concurrently randomised cohort, and that's why we sort of don't discuss these concepts within the context of traditional designs. But as we move into some of these more elaborate designs, we can have more than one concurrently randomised cohort because patients are potentially subjected to different chance mechanisms. But what will be the case is that all of the participants belong to one and one only concurrently randomised cohort. And so this concept is going to be key to defining our randomised comparisons. Incidentally, this was sort of expounded in some depth in a paper a couple of years ago that I wrote with the Ascot group, and using the Ascot trial as a case study in defining some of these concepts. All right, so using the concept of a concurrently randomised cohort, we then want to get to a point where we have a rigorous definition of concurrent and non-concurrent controls. And that definition is as follows. So this is a definition of a non-concurrent control for somebody who is in an experimental treatment arm. So for a participant who's in an experimental treatment arm, a non-concurrent control for that participant is a control arm participant who is not a member of the same concurrently randomised cohort. So remember, all participants are a member of one and one only concurrently randomised cohort. And the control participants who are not in the same concurrently randomised cohort would be called non-concurrent controls. And the complement of that is that a concurrent control is a control arm participant who is a member of the same concurrently randomised cohort. Notice that this definition doesn't mention time at all. So the time of randomisation is totally irrelevant to this definition of concurrent controls. And in particular, I would contrast the concept of a concurrent control with the concept of a contemporaneous control. So a contemporaneous control is a control participant that was randomised at the same time, using some definition of what we mean by the same time. But that is not the same as a concurrent control. And in fact, you could have a contemporaneous control who is not concurrent. And I had some examples of that with the Ascot trial earlier. And the reason why concurrency and not contemporaneity is the important issue is because the randomisation is being conducted within the concurrently randomised cohort. So it's the concurrently randomised cohort, regardless of time of randomisation, that is delivering us the randomised comparison. And that then allows us, I guess, somewhat trivially to focus in on what we mean now by a randomised comparison. This is a concept that we sort of all have an intuitive notion of, but just to define it rigorously within the context of designs where we have these other sorts of control groups, a randomised comparison now is a comparison of treatments within a concurrently randomised cohort. A comparison of treatments between concurrently randomised cohorts is a non-randomised comparison. And so we've firmed up our notion of what we mean by a randomised comparison through this concept of a concurrently randomised cohort. And it's those cohorts that deliver us the randomised information. Of course, within the same study, we might have both of these sorts of comparisons. We might have randomised comparisons and non-randomised comparisons. And so a valid statistical analysis method is going to involve, first and foremost, preserving the information from the randomised comparison. So this is why it's so important to be careful about how we define a randomised comparison, because we want to make sure we can extract the randomised information from the study. And then once we've done that, we can then possibly, or even possibly not, combine that information with the non-randomised comparison. So we can make a decision about whether to use the non-randomised information or to stick just with the randomised information. And then if we just revisit our examples, we can see that within the context of the Ascot study, what we have then is 3 concurrently randomised cohorts, 880, 482, 194. And within the remap-cap example, what we have are 3 concurrently randomised cohorts, 1383, 1277, and then a very small one at the end of just 24 patients. And each of those 3 cohorts yields us a randomised comparison, and we need to extract that information from the study and then decide what to do with it. Now, in the remap-cap example, there's no other non-randomised non-concurrent controls. There's no other information to bring in there, whereas in the Ascot trial, there is potentially. So we've got, for example, a randomised comparison between therapeutic dose and low dose, and we need to make a decision about whether to bring in the low dose information from the other concurrently randomised cohorts. Just by the way, just to see the effect of these concurrently randomised cohorts on the baseline characteristics. So you remember earlier I showed you this side of the Ascot table where we had, for example, a very big difference in vaccination status from 15% to 54%. When we break it down into concurrently randomised cohorts, then those differences essentially fall back to be within the range of chance variability of the randomisation scheme. So we have one concurrently randomised cohort that has little vaccination, medium and higher, and we have that balance between the groups, at least within the range of the chance variation of the randomisation system. Now, I said I won't sort of go too deeply into the methods. I'll mention some of these methods and then just give you a brief illustration of conceptually what's going on with some of these methods when we use them. So as we said, you know, we're susceptible to bias here, so we do have to be careful about the statistical methodology that we use. There is various techniques that can be used. I'm going to illustrate to you this concept of information borrowing at a conceptual level in a minute, but there's other ways that we can do it as well. Adjusting for time period, adjusting for randomization cohort. There's an analog of network analysis, network meta-analysis that we can use. And there's also various sorts of matching techniques, propensity scores, synthetic controls that we can use. And there's a whole vibrant field of statistical methodology around that. I'm trying to illustrate some of the underlying concepts here. And fundamentally, the methods that we use to analyze these sorts of control groups depend on whether or not we have a concurrent reference group. We could either not have a concurrent reference group or have a concurrent reference group. And what I mean by that, in the Ascot example, for example, the yellow and the purple. So if we think about comparing the blue and the pink here, that's a bit like a historical control comparison in the sense that there's no overlap between these two. Now, they happen to occur in the same study in this case, but it's a bit like a historical control example. In that particular example, the yellow and the purple provide a concurrent reference. So we have a randomized comparison between blue and purple, and we have a randomized comparison between pink and purple. And so therefore we have that common reference of the low dose heparin in this case that allows us to link the blue group with the historical group. And the analytical method that we'll use is much more powerful when we have that particular situation. So that's why I said earlier, in terms of the historical controls, it's much more powerful when we have a control group within our study, even if it is a small underpowered control group, because it provides that common concurrent reference group that we can use in the statistical analysis. And it's essentially like, if some of you who are familiar with indirect comparisons, mixed treatment comparisons, network meta-analysis, it's the same sort of concept. Usually when we have non-concurrent controls, we will have that concurrent reference, whereas with historical controls, we may or we may not. So just to give an idea of how we use that concurrent reference group when we analyse either non-concurrent controls or historical controls that have that reference, I'll use the Stampede example again, Stampede trial as an example. So here I've got three groups. So this is prostate cancer, androgen deprivation therapy, the combination with docetaxel, and the combination with abiraterone. And suppose we're interested in comparing these two combination groups. So we have a randomised comparison between the two combination groups, the purple and the green, yielded a hazard ratio of 1.16. And then we also have various other randomised comparisons between the combination and the ADT monotherapy, hazard ratio of 0.63, between these two, 0.72. And then in the earlier part of the study, we have a hazard ratio of 0.81 between the red and the green. All right, so we have a randomised comparison between the two combination groups, but we might ask the question, can we use this information from the non-concurrently randomised patients from earlier in the study? The way in which we do that is to essentially borrow the information from that part of the study by, if you like, if you want to think about it, like imputing from this randomised comparison a hazard ratio of 0.63 between this counterfactual grey group that we didn't actually even observe, but assume that we would have got the same hazard ratio as we got in the randomised section of the study. And then what we have is a hazard ratio of 0.81 compared to a hazard ratio of 0.63. 0.63 over 0.81 is 0.78. And so essentially what we've done is impute a hazard ratio for the comparison between the two combination therapies of 0.78 in that earlier part of the study. And that essentially then gives us two comparisons between the combination therapy groups, and we can look at comparing those two and indeed aggregating those two. And that's what we might consider in an analysis that brings in that non-concurrently randomised information. And so we would go from the direct randomised comparison of 1.16 to this aggregated comparison that borrows the information from the indirect or non-randomised information. And that illustrates two things. One, the sort of process of borrowing the information from the earlier part of the study. It also, though, illustrates the danger, I think, of using this non-concurrent information, because in this particular example, in the Stampede trial, and by the way, the Stampede study restricted its comparisons to randomised comparisons, so they didn't do this. This is just an illustrative analysis. They relied just on the direct comparison here. But it shows that you can have quite big inconsistencies between the randomised and the non-randomised information, which might then lead to that bias that we were talking about earlier. So overall, just brief high-level summary of the sorts of things that you can do. You can do crude pooling and just throw all the control group patients in together. That's called simple pooling, but is virtually never recommended. In situations where we don't have a concurrent reference group, we can use matching type techniques where we try to construct control groups that are in some sense matched to the group that we've done the intervention on. We can restrict to the randomised analysis, and for primary analyses and late stage confirmatory trials, that's the method that I prefer. We can do various sorts of adjusted analysis, adjust for the time trends, adjust for the cohort differences, use this sort of network of indirect and direct comparisons. We can test then pool. So in other words, in that previous example, we would test whether there is an inconsistency. And if it is inconsistent, then we wouldn't pool. But if it is consistent, then we would pool. And there's also the concept of dynamic information borrowing, where we essentially use a weighted average of the randomised and the information borrowed analysis, with the weighting determined essentially automatically based on that measure of inconsistency, and that's probably an ongoing research topic. All right, I might skip over, there are some implications for the database in these sorts of scenarios. I might skip over that because it's sort of a tangential thing. And then I'll just cut to a summary of the pros and cons, which really come down to this bias versus information trade-off. The risk of bias from introducing non-randomised information into our analysis, balanced with the potential to increase our information. And really that stampede example illustrated that scenario of the risk of bias. And from my perspective, I think increasing the information earlier on in the development stage makes a lot of sense. As we get to the confirmatory stage, it probably makes more sense to rely on the randomised information only. So key points from the discussion. So obviously historical and non-concurrent controls do not provide a randomised comparison, and as non-randomised comparisons, they are therefore susceptible to bias. Randomised comparisons involving concurrently randomised cohorts are preferable for the primary analysis. Non-randomised comparisons involving historical or non-concurrent controls may be useful to increase information in resource-limited or rare disease scenarios, and historical and non-concurrent controls require special statistical techniques. And finally, just to end with a guiding principle, we're talking about non-randomised information here. Whenever we do have randomised and non-randomised information, however we ultimately analyse it, it's important to transparently present the different contributions from those two sources so that their consistency can be assessed. And coincidentally, that's the topic of one of our recent papers that deals precisely with that topic and promotes that guiding principle. Well, thanks for your attention, and happy to take any questions.
Thanks very much, Ian, for a wonderful presentation. As usual, sort of illuminating concepts have been around for a while. You see other angles and presenting them in a very clear manner. I'll open for questions. We don't have any in the Q&A, so we'll open it up just for people to, should be able to put up your hand there. But it was really refreshing to see there was not a word about frequentist or Bayesian there. And as you said in that very last slide, we're talking about fundamental principles here, and sometimes it's easy to get lost in the details of borrowing strength by various priors or whatever, but that's not a relevant.
I think they're concepts that apply to both paradigms, and so that's why it sort of wasn't getting bogged down in the different differences between the two.
We have one question from David Patterson. Can you let us know how regulatory agencies such as the FDA assess the use of non-concurrent controls?
Yeah, well, there's a couple of guidances from the FDA that relate to this. There is a guidance on the use of master protocol designs, which include platform designs. And I think that's still at the draft level, but there's a very clear recommendation in that draft guidance that the primary analysis should be based on randomised information. So the use of non-concurrent controls in FDA regulatory submissions from master protocol designs would be restricting this to sensitivity analyses and secondary analyses. There's also a guidance on historical controls as well. So obviously they do entertain the use of those designs, but clearly have a preference for randomised comparisons.
Yeah, thanks. Someone just sent me a text about it. Yeah, so people can raise their hand if you want to for questions. In the meantime, I'll just ask another question there. So This is to your Stampede example with the what's called concurrent reference. There seemed to be an implicit sort of treatment effect homogeneity over time assumption that was being made there, where you could transplant that hazard ratio of 0.63 or whatever it was back into the earlier time period, even though the comparison with, I've forgotten what colour the other arm was, changed from 0.7 to 0.8. So it's almost an assumption it's the stability, the control arm was stable, and it was the other arm that caused that change in the hazard ratio.
Yeah, so that is a fundamental assumption, isn't it? That the treatment effect is remaining constant over time. So those information borrowing techniques can take account of background risk changing over time, but taking account of the treatment effect changing over time is another level of complexity, and is essentially what you're dealing with there is an interaction between the treatment effect and the time. That's what the inconsistency is in that context. And that needs to be assessed and tested for, I guess, before you can use these techniques.
And either Bayesian or frequentist methods can cope with sort of uncertainty about that by various components or priors. So,
yeah.
We've got another question here. Given this, this is from Christy Robledo. Given this research, how do you view adaptive randomization schemes that amend the ratios based on the winning treatment arms?
Yep.
Do they need to be cautious, perhaps using a threshold before undating, before updating, yes, rather than updating at each interim analysis?
Yeah, so I mean, a good example there was the remap cap example. So look, you can use these techniques, but the important thing is to extract the randomized information from the cohorts that have the same randomization probabilities. That's what the randomized information is. And one of the key issues here is the frequency with which you're adapting the randomization. So in that remap cap example, there was just a couple of adaptations. In some designs, we have very frequent adaptation, and that really limits the amount of randomized information that we actually have, because the randomized groups, the concurrently randomized cohorts, become very small if we adapt frequently. And if you take it to its logical extension, if we're adapting after every patient in the study, then I would argue that that is now no longer a randomized study at all, because we have no concurrently randomized information from that. So I'm very, I very much prefer with these adaptive randomization schemes and all adaptive designs that the frequency of adaptation is limited, that we don't have very high frequency of adaptation, because that reduces the amount of randomized information that we have.
I think Christy was also saying about whether you need to have a sort of substantial run-in period, let's say equal randomization first.
Yeah. Well, certainly, because the problem with a short run-in period is that you can, due to chance variation, you could easily start going in the wrong direction in terms of which treatment is better, and therefore which treatment you're favouring with the randomization. So having a reasonably large run-in period, which is kind of related to the point that I was just making, which is that we have reasonably sized concurrently randomized cohorts, both in terms of the run-in period, but also in the subsequent periods as well.
I've got another question from Mason here. Thanks for the great talk, Ian. In the context of the network meta-analysis, does indirect comparisons, are they considered to be fundamentally non-randomized, given your definition, or it depends on the amount of
indirect comparisons are non-randomized comparisons, but they're non-randomized comparisons, at least, that are based on randomized comparisons. So if you compare the example I gave with the historic controls, if you've got, a historic control group and a currently treated group, that comparison is a non-randomized comparison, which is at a lower level than the indirect comparison that you have in a network meta-analysis, because at least you're comparing two randomized comparisons to get an indirect comparison between the treatments that you're comparing. And it's the common reference group within the indirect comparison or the network meta-analysis that gives you a little bit more methodological integrity. But I would still refer to it as a non-randomized comparison.
So there can be indirect, I think in one of your examples, just following on from that, indirect comparisons in two completely separate time periods.
Yep.
Yeah.
Yeah. Yeah.
And then the assumption of the treatment effect staying constant over time is really fundamental then.
Yeah, and that example where it was two separate time periods, that is fundamentally a non-randomized comparison because there was no concurrent randomization to those two treatment arms. So it has to be viewed as a non-randomized comparison.
Let's see. No further questions at the moment. Okay, so I'll just ask a question. So what trials have you been involved in directly with the analysis and used the various methods? In one of your last slides, you gave about six or seven different approaches. Have you implemented any of those in practice with any of the trials you're involved in that are sort of ongoing?
Well, the best example is the Ascot study that I was on the statistical committee for, and probably there's people on the call who were also involved. I can't see all of the attendees, but they'd be on the call as well. And I guess the primary analysis for the Ascot trial, and that was done in a Bayesian framework, the primary analysis, but the primary analysis for that trial brought in the non-concurrent control information. And I guess that subsequent paper that we wrote was really an attempt to delve down more deeply into the separation of the randomized and non-randomized information. And so that's where a lot of these concepts were born. I've also subsequently, and I wasn't involved in the Stampede trial, but we've done some analyses of the Stampede examples to illustrate some of these concepts with here at the Trial Centre as well. And the other methodology, I guess, that we've explored using some of these trials is the cohort-adjusted methodology. So typically, the primary methods that you'll see for dealing with non-concurrent controls are based on time adjustments, and we've used cohort adjustments as well within that context. So yeah, I mean, some of those methodologies that I listed. I mean, to be honest, I haven't done a lot in relation to the historical controls, synthetic controls, that sort of thing, but I'm sort of just aware of the methodology that's used there.
Right, thanks. We're pretty much out of time, but I can't resist the opportunity that Rory Wolf alerted me to something that was rather tragic occasion of a tornado off the coast of Sicily that capsized a luxury 15-meter yacht, and it was called the Bayesian, because the owner apparently made his fortune from what's described in the media, the statistical scheme called Bayesian.
They were all saved by a boat called the Frequentist. Is that what happened? No.
6 still missing. But, okay, everyone, let's thank Ian for a wonderful talk and some very interesting questions at the end, and great responses. And I think we've all learned a lot more about what to be looking out for when we're comparing with non-concurrent controls. There will be a short survey when you exit the WebEx link, so please do complete that. And a reminder, Kate, the next Extra Sting event, there'll certainly be a breakfast meeting at the ACTA annual scientific meeting in November, so please do look out for information about that. And thanks to the ACTA admins for organising the session. So I think we'll sign off now, and thanks again very much, Ian.